So, yesterday we finished our conversation on cellular agriculture and precision fermentation, right? When we were discussing the topic of cellular agriculture, we talked about the acellular productions or product productions. We also talked about the cell biomass being itself a product. So, when we were discussing about the acellular production, I talked about transforming the host or providing the capability to the host that it can produce the product of interest, right? Now, how do we do that? How do you transform your host or provide it with the capabilities to produce the product of your interest? You have to make some modifications, right? It's like asking if you want to change someone's personality, you first have to understand the person itself, right? So, keeping that in mind, so this part what we just said, like to change the personality of the person, you need to know the person itself. That's what sets the basis of all our conversation that we are going to have today. It would be more about understanding what happens or what changes we can make to the bacteria or to the host organisms, specifically like yeast. Bacteria would be the E. coli, and what are the tools that are available with us. All of this that we are going to discuss fall under three different topics that what we have: the synthetic biology, the systems biology, and then you have the enzymes and the engineering enzymes. So, we will start with synthetic biology. Now, in my own personal viewpoint, synthetic biology actually encompasses all the concepts that we should be discussing today. If you are talking about system biology, it has the parts of system biology in it. If you are talking about enzymes, it is going to have the parts of enzymes in it. So, you might be getting a bit more of that feeling that you already are able to understand the enzyme engineering or engineered enzymes in here, plus what the system biology exists. Now, what you see here is the definition for the synthetic biology. So, synthetic biology is defined as the design and construction of new biological entities such as enzymes, genetic circuits and cells, or redesign of existing biological systems. So, as we started our conversation was that if you want to make a change into a person, you need to know the person first, right? So, if I know the host, or if I, let's say if I have to make modifications to E. coli, I want the E. coli to be able to produce a specific product. If I know how the E. coli functions, what is the metabolic pathways that are followed within the E. coli system, I should be able to tweak the E. coli or tweak the metabolic pathways in such a way that I can get the product of my interest. Do you agree to that? How do we tweak it falls within synthetic biology. What are the tools, what are the engineering tools that actually we use will fall under this particular topic. Now, it is different from the traditional molecular and cellular biology, as the focus is on the design and construction of core components. Because we are only focusing on the design and construction of the core components, it differs from the molecular and cellular biology. So, we will be looking at the metabolic pathways, part of the enzymes, we will also be looking at genetic circuit. So, whenever I keep saying genetic circuit, it's like you have the genome, you know what the genetic code is and what each and every gene is going to represent, like what it will translate into. So, if you know that there is a gene sequence, let's say, and every section of the gene is going to translate into one specific protein. So, you will get 3 different proteins. Now, my point is, what if my interest is that this particular genome should only be producing number 3, or should be producing number 3 more than the rest of the other proteins like 1 and 2. So, if I can repress these sections of the gene and only allow this section to translate, I would be only getting my product of interest. So, there are ways to do it, right? How do we do it? Those are the tools that we are going to discuss. On the left hand side, you will see the basic the roadmap that is basically used in the synthetic biology. The DBL or DBTL is your design. What is your B? Build, test and learn. As an engineer, when I am talking about as an engineer, engineering involves designing things, right? So, we always design first, we build it, a prototype, we test the prototype, we learn from the prototype, and only then we will go into the bigger format of it, right? So, that is what exactly used at a molecular level. So, we have taken a macro level idea towards a molecular level idea. Now, so the goal is to engineer cells to produce something useful, and I will be showing you some of the examples. We will be discussing a bit more in detail about those examples. So, for example, if we have to look at the bacterial production of insulin, and if we are using host, you remember when we were discussing the host selection is a very important parameter when we are looking at production of a specific product of interest. Now, what was the criteria for the host selection? Can you repeat some of those criteria? Non-pathogenic.
Able to put, it should give more quantity.
So, you should be able to produce more
more
of your product and ease of downstream processing or ease of extraction or ease of recovery. What else?
Understanding of different metabolism pathways.
So, if I had to say it in a layman way, it would be that you should be aware of the host capabilities in such a way that you can make the modifications that are required.
And cost effective.
Cost effectiveness will come into picture only then, right? If you are not aware what the host can do, it will be a very costly affair, right? Good. So, I am happy that you listened to the things that we were discussing yesterday. So, same thing, now the same concept. Now, let us say if you have to produce insulin and you are using E. coli as your host, what will be the very first thing that you are going to do? You are going to design a genetic circuit of the E. coli or of the insulin production. So, what you have is, let us say, so this is, please excuse my drawing, if this is your E. coli, this is your genetic material, your DNA of the E. coli. Okay? Now, there are two ways of producing your product of interest. Now, if the insulin, now where do you get your insulin gene or the gene that is going to translate into insulin? Where will you get that insulin, the gene of insulin from? What would be the source? Who requires insulin? Humans, right? If somebody has diabetes, insulin is basically produced to break down the sugar, right? So, you need the insulin. So, you will get that genetic information from the human genome that has already been looked at, right? So, you can take out the genetic sequence from there. Now, you can take that genetic sequence and you can pass it on into the E. coli using a vehicle. Now, that vehicle is called a plasmid. So, this is your plasmid. Now, what is the other unique property of the plasmid? So, plasmid contains the sequence of interest, so that it can generate more of it, right? As it will replicate, it will keep on generating more of that particular sequence, and then eventually that sequence will translate into the protein, if that protein is my protein of interest, when the E. coli is growing, right? Now, what other properties? Now, do you think every E. coli that I have my plasmid into will be able to run the plasmid, or the plasmid will work in that E. coli system? I might not be able to frame my question properly. So, what I am trying to say is, let us say if you have 10 E. colis, so you have provided the plasmids into all, right? Now, will all of them be producing insulin at the same efficiency, or will they have different efficiencies? Like some of them might have lower efficiency, some of the strains might have a higher efficiency, some would be like mediocre. Which one will you choose? And how will you choose? It depends on the plasmid. In what way? Like, what is the structure of a plasmid? It's circular. I was not asking that. Like, what would be the component of the plasmid? So you have your zone of origin, and then you have your antibiotic resistance. Why do you have this? Screening of what?
Yes, obviously. So if we have antibiotic resistance, then we can grow it on, and we will like get the bacterias in which we have our insulin, and in those which we don't have our insulin.
So can plasmid also be used for selection purposes? So is that a tool to add selection properties?
So can you repeat your questions?
So if you have the antibiotic resistance gene there, so any of the bacteria that will actually have the plasmid and that has replicated the plasmid within it will be antibiotic resistant, right? Or it will showcase antibiotic resistance.
Yep.
So if my protein of interest or the insulin is being produced, it will also be capable of being antibiotic resistant. Not all bacteria would be doing that, but that one would be. So if I am only interested in the one which is producing my insulin to the maximum, can I use this as a selection criteria?
For maximum we can't use it. Like everybody will be producing some or the other amount of the insulin. For maximum property we can't use
We
that.
cannot use that.
Yeah.
But what if like one which is not producing, one which is producing? We can do that?
We can do that.
Right? Very good. So for this type of selection, it cannot be used. But when you are selecting it from the microbes that don't have the plasmid or they're not producing your insulin, you would be able to use the plasmid as a selection criteria, or that antibiotic resistance property that is provided by it. Now, so you have your host. So either you are going to add the plasmid to provide that insulin in there, or you can make a cut within the genome of the bacteria or the E. coli and add your gene of interest to produce your insulin. Which one do you think is much more effective and easier? So either I make the changes to the genome of the E. coli itself, or if I'm adding that external DNA strand using a plasmid in the E. coli, which process would be much more effective and which process would be much more easier to do? Using a plasmid would be easy. Why?
To some extent, we are sure that it will be producing the insulin.
Now you have answered it, but you have answered it in a very different way. I like the answer. Actually, it's correct. So the other thing is, like, when you are incorporating the specific genetic material in the E. coli DNA itself, there is no guarantee that it might get transcribed too. It might be in a sequence that that material or the genetic material never gets transcribed. Not all part of the DNA is transcribed, only some part of it does, right? So plasmid, having a plasmid guarantees that it would be there, but having it in the DNA sequence itself doesn't guarantee that. So the efficiency or effectiveness might be less, but there is no way until unless you do experimentation to actually state whether it is better or not. So there are two ways of doing it. Either you add the plasmid or you can add it into the DNA sequence itself. So now you have to build the genetic circuit and put it into the bacteria. So I have built the genetic circuit like your plasmid, which is your genetic circuit, and I put it inside the bacteria. And then you test how much insulin is made or how much insulin is being produced. And once I have optimized the strain that is actually able to produce most of the insulin, I can take that strain and start growing them in the fermenter. Now that is exactly what this particular flowchart or the set of diagrams are representing. So you have your human insulin gene DNA, you have your plasmid loop of the bacterial DNA, which is there in the E. coli. Now plasmids were actually found in E. coli. They are separate to the DNA of the E. coli. and these plasmids were the ones which were providing some unique properties to the E. coli, and that's where the interest for us as scientists came into for the plasmid, that hey, we can use these as materials to transport. It can be an amazing vector. So now you have inserted your gene of interest for the insulin into the plasmid loop, you pass it on to the bacteria, you grow it, you select it, you put it into the fermenter, and you start producing the insulin. Now that once insulin is produced, or enough insulin is produced, you can harvest it and then purify, and then you get your medicine, your insulin. Okay? Now this is called your recombinant DNA technology. Technically, this is your recombinant DNA technology, and also an important part of precision fermentation. Now, synthetic biology actually encompasses the tools that were used to make that change of how to produce that insulin. What were the tools that were used to move the, or to cut the genetic sequence for the insulin from the human genome, put it into the plasmid, put it back into the E. coli cells, let it grow. The growing part would come into the fermentation aspect, but the rest of the things before we get into that is your synthetic biology. But if you don't understand the metabolic pathway, or if you don't understand the microbe itself, it will be very difficult to make the changes that we wanted. Now that part, understanding the microbe as a whole, what happens within the microbe, would come under the systems biology. And if instead of insulin, I was, let's say, developing or producing a new enzyme, or I was making modification in the DNA sequence of the host to produce an enzyme of my interest with my capabilities that I like that enzyme to have, that would be your enzyme engineering. All three technically can be considered as one topic, but there are three different tools that are used. Now, so the approach that we use with respect to the design, build and test is the designing, which is your biological circuit or a system for a specific function using computational tools. The build consists of a system by assembling, editing and installing your genetic material. So, that is where your plasmid comes into picture. Then you have your test, the system functionality, that once you have added whether you are able to produce the insulin or not, that would be your test. And once you have done that, you put it into your fermenter system, you can learn how the system is performing, what sort of modifications you need or what optimizations you need to produce more and more of it. Now, this is your roadmap for your synthetic biology. Now, let us look into the goal, how the same thing that we had here, the design, the build, the test and learn, how we implemented for our insulin. So, the very first part where we are designing the insulin precursors. So, designed by finding the gene of the human insulin precursors and optimize model for the E. coli expression, and then you move it on to build the gene sequence including the insulin precursor genes through synthesis and assembly. And then you test your circuit in cells by measuring the amount of insulin that is being produced, and then learn how the system works and at what optimization is necessary for growing the insulin in the fermentation process. So, your DBTL can also is part of your precision fermentation, but before that, so where we were looking at starting the culture, so preparing the culture, the host, and then moving it on into the lab scale 5 liter, 10 liter. So, you will have the shaker flasks. So, all of these experimentations are done first in the shaker flasks to make sure that they are working. Then you do it into a small scale, that will be 2 liter or 5 liter bioreactor, and then you will do it in a pilot scale, and then you will move it on to a commercial scale. One of the best examples that I can actually give with respect to application of synthetic biology, and an amazing application of synthetic biology, would be the anti-malarial drug artemisinin. Now, artemisinin is produced in a plant, Artemisia annua, that particular species, which is throughout the throughout Asia and Africa, and it is used as a tea, and its pressed juice is used to treat malaria and related symptoms like fevers and chills. Its active ingredient is your artemisinin, which has been developed as an antimicrobial drug, anti-malarial drug, and is used worldwide. If I have to get this artemisinin to make my drug from the plant itself, what would be the process that you will follow? The process is there on the slide, so you can just explain me that, but what would be the process that you will follow? You're going to grow the plant,
and extract it from
then
it.
you have to come up with the process or extraction process,
Chop
the
and
protocol,
like this.
the chop it, pre-treat
Clean
it,
it, pre-treat.
and what else? So pre-treatment, okay. So grow, harvest.
Post harvest.
I'm not going to do chop.
Okay.
Okay. Chop,
Yeah. And then clean or something.
chop can include the cleaning part also. Okay, let's say pre-treatment,
Pre-treat, much more important one.
and then you are going to do the extraction.
Yeah. Either powder form or something. Like
And once you, okay, why chloroform?
no, powder.
Oh, the powder form. Okay. So extraction, and then you are going to put that into a powder form. So for that you have to dry it, right? And are you using a conventional oven, or you are using a freeze dryer?
Based on the budget.
Based on the budget, okay. I will define the budget here, because we are going to spend some time on this particular slide. So,
yes.
okay, so you have your budget, you have your drying, you have your powder form, and then you do what? You give the powder to the people to eat?
You can give for the next with the microbial we can use as the feed. Feed for.
You're using this powder for what?
So for the next stage, for the
for the next
fermentation.
stage.
For the
Oh, the thing that you are saying about. No, artemisinin comes out after extraction, so this particular powder can be used as is, like, so that's. You have so many examples coming out from Patanjali.
Actually, I gave it with respect to my work, what I am doing. So that's why I gave the same.
Okay, no, I realized that, but I'm just so you have the chop, you have the pre-treatment. That's a very mechanical way of saying it. I really like it. Now, somebody can explain me in the biological or biotechnological perspective what the chop will mean.
I'm from food engineering background.
Okay.
And I will try my best. Okay, so, for example, if whatever the antimicrobial drugs, there will be anti-malarial drugs, there is a lethal dose to like a specific dosage is required to destroy the like get the effect. So first of all, for whatever drugs are which is available, which is in the encapsulated form or compressed form, that is available in the market. So accordingly, we can extract it and we can encapsulate it, and the controlled delivery can be done. And it should be stable in the pH in our gut, and it should be like wherever it is required. So I thought like definitely it should enter into the bloodstream. So definitely it should be stable until large intestine.
Okay.
So it should be stable. Like we have to encapsulate it in that form to deliver into our drug. So most of the antimicrobial forms, I think it's in encapsulated form is available. First of all, pelletization. After that, we can encapsulate it.
No, so you're talking about downstream. Once you have the powder, I was asking if there's a way that you can explain what chop was. So you had the grow, you have harvested it, you cut down the tree, macerated it, got a powder out of it, of the tree itself. So and then you are doing pre-treatment. Pre-treatment means reducing the moisture content, right?
Yeah.
Or by making a fine powder so that you can increase the surface area, because to improve your solvent extraction efficiency, you need to have a very high surface area, right? So you will do that, and then you will do the extraction process. Now, what would be the solvents that you will use for extraction?
It depends upon even different solvent having even different polarity. So this particular
if it is a polar compound, you are going to use a polar solvent. If it is a non-polar, you are going to use a non-polar solvent, right? Let's say if it is a non-polar compound.
we will be using non-polar
Like? What would be the non-polar solvent that you will use for extraction purpose? Go ahead, you can answer. It's okay. There is no wrong answer in this particular session, so please go ahead. Chloroform, acetone, hexane. Okay. Diethyl ether or else? So almost all of these are your non-polar solvents. Are they good for you? So they're always going to leave a residue, right? When you're doing an extraction with chloroform and acetone and hexane, they're going to leave a residue on your powder after the drying process. So what are you going to do then? You can use novel techniques and other things for the extraction purposes, but when you're looking at it from the practical perspective, you might not be able to go into microwave assisted extraction or ultrasonication. I'm not going there. That's not the topic of our conversation. I can give you a 4-hour or 10-hour lecture on that. But my question here was that once you get the powder, you're basically looking at a highest purity because you're going to use it as a drug, right? So you want to get rid of any traces of any solvent that was basically used. Now this whole process is a very long process, and the solvents that you are going to use are also not very good for your health, and you will need a very costly downstream processing methodology that will get rid of the traces of these organic solvents, and you will get your product of best quality or the highest quality. Do you agree with that? Right? So practically it's possible, but I will not like it, or I will not like to use it for let's say mass production. What I would like to have in that case is to identify which part of the plant genome is responsible to produce this artemisinin, and then I can use that, put it into a genetically modified E. coli or a yeast or a fungi, grow it, produce it on a larger scale, right? That would be the interest. And that is exactly what people did, because malaria is a major issue in Africa. It's also a major issue in India. And at one point of time, there were very limited availability of drugs, and the drugs that were available were very costly. So synthetic biology actually provided the possibility to develop artemisinin at a very cost effective way, or in a very cost effective way, and it made its availability across the globe, and now that is also used as a very first line of defense. So we are using this as the first treatment that you give to make sure that the person is cured of malaria. So one of that example now would be that the production of amorphidine, which is your precursor, and its conversion to dihydroartemisinic acid, which is again the precursor for artemisinin. So amorphidine is being produced in the yeast. Now yeast as a source, because before this they did in E. coli. When they did that in the E. coli, the overall yield was less. They made changes, they looked into other hosts, they found yeast, they made modification to the yeast, they got much better yield. So that's how the host selection will also change, because you sometimes, it's not that you can decide on the day one itself that, you know what, this is the best host that I can use for my genetic material to grow. You might have to do some trials onto it. So you will do with the E. coli, you will see whether you are able to improve the overall functionality and production of your product of interest. If it doesn't work, then you try. If you have understood the metabolic pathways in the yeast, and if you can make modification into the yeast, you will start growing into that, because the fermentation process will change. What sort of fermentation system do you think you will need for the yeast? Because in the later courses or the later lectures that we have tomorrow and day after, we are going to talk about batch reactors, We are going to talk about the fed batch reactors, aren't we? So, we are going to talk about these type of reactors and how to design these type of reactors. I am not saying physically, but mathematically. So, what sort of bioreactor you are going to use for which type of host matters the most. So, when you are going to use yeast, and for that you only need the input so that you can produce your product, I can use a fed batch, where I can only give the input, there is no output. In a batch reactor, there is no input, there is no output. Once you put everything in, you close, and that's it. It's like when you cook rice in a pressure cooker. You put the rice, you put the water, close it on the stove, done. And then you wait it to whistle. That's your batch reactor. Good example. Now, same rice, you added less water initially, but it's an open pan. You'll add more water as it is boiling, right? So, technically that becomes a fed batch. So, you'll add the nutrients for the cells to grow. So, for the yeast we generally use fed batch. So, this one was done on a fed batch reactor, and what you will see here, this particular diagram here, and I really insist if you can read this particular paper, it's a really interesting paper. They did a genetic modification into the genome of the yeast, and they added the sequence, the genes that were necessary for producing the precursors, so that they can produce the artemisinin at the very end. And what they found is, like, as compared to their previous cultures, or which were used earlier, the amount of artemisinin that was being produced was pretty high. The amorphidin, and also you can look at the artemisinic acid, is pretty high. So, your Y151 is your generation 1, this is your generation 2. So, there were two different generations of the microbe and the plasmids that were being used, or the processes that were being used within those microbes to grow and to produce these precursors. So, in the generation 2 you get a much better yield. So, that is how we keep on improving our productivity. We have to continue. So, we can start with the E. coli, we might get to a certain level, we will then change it, we will try to another host, we might be able to get something else. As part of your GAN series, you are going to do 2 case studies. One is based on a plant protein. We are going to have some form of discussion today. I am going to tell you which product I want and what host I would have chosen for it. I would like you to find out by yourself. You can work in groups. What source will you choose? Like, what host should be there, and what will be the process, and where will you get your sequence from? How will you get that sequence from? All of that will be discussed in this class today. So, in the synthetic biology, the systems biology, and the enzyme engineering. So, I will give you the background for it to build on your case study. So, what? It's a good example, right? So, like the producing the malarial parasite, sorry, not a malarial parasite, malarial drug. Please excuse me on that. You are not producing the malaria parasite. But do you know how the malaria parasite actually works? You have heard people get malaria, but do you know why do they get malaria? Or how does that Plasmodium falciparum, which is the parasite, impacts? So, when it comes inside, what does it do? Where does it grow? I am not able to hear you, so I am so sorry.
RBC?
In your red blood cells. Have you heard sickle cell anemia? What is sickle cell anemia? Your red blood cells are in a sickle cell shape, right? And that is caused because of a genetic mutation. People having sickle cell anemia cannot contract malaria. So, a genetic modification can be good for you, it will kill you anyway, but not with malaria at least, right? But the point is, these genetic modifications are very, very important. They are very natural. So, microbes go through genetic modification in a very natural way. So, let us say if you want to produce a specific genetic product or like a protein, but you want to make sure that it is done in a very evolutionary way. So, you will make a small modification to the DNA sequence, you will let that mutate from one generation to the second generation to the third generation, and you will keep on checking if your protein of interest is being produced and in what quantity. And the moment you reach the strain that is actually producing the protein of interest in the highest quantity possible, you choose that. But that has been a very evolutionary way of making that modification. It's a very tiring process, it's a long process, but it's a good process. Okay? We will be discussing that later. I'm actually going way ahead of many of the topics sometimes. Another example, how many of you are actually having a water bottle in your hand, a plastic bottle? Do you have a plastic bottle? Just like the one which I am going to showcase right now. So, like this. Do you have this? Anyone has that? Or have you ever seen any marking at the bottom? You will always see a marking like this on a plastic bottle, right? Now, terephthalic acid is the precursor which is basically used to make PET. So, terephthalic acid is a precursor which is basically used to make your PET, and this PET is mostly used for your making the bottles and the plastics, right? Now, if you are looking at the conventional process, you are going to see that it is using cobalt, it is using manganese, those are heavy metals. And you would have heard about heavy metal pollution, right, in the soil, in the water. So, if you are looking at a process, a chemical process that are using these metals, you do not want to use them because you would prefer to change it. It is not environmentally sustainable process. So, we have to come up with something else. But if there is a way for us to produce this PTA within a microbe, I would love to do that, because I can produce as much as I want or even more. But what you have, like in the conventional process, in the normal regular processing, you are producing in bulk, and that is the reason why it is only costing you very less. It is only $1 a kg. So, the cost of that is pretty less because you are producing in bulk using chemicals. Now, the same thing was done using microbes, and the efficiency that they got was approximately 6 grams per liter, which was equivalent, to be honest. It is very equivalent or similar to what you would have got in a conventional process. But when you see the diagram on the left, it looks very, very complex. But if you have taken a course in biochemistry, you would be able to identify some of the components within that, right? Can you identify those for me? You have, it's actually saying you have your glycolysis cycle, you have your TCA cycle, that's your biochemistry 101. That's the very first thing that you study in biochemistry, right? Now, why do we need to have a glycolysis cycle and a TCA cycle? Who has this cycle? Here, in this particular situation, who has this cycle? Where is this cycle present right now? Within the microbe, within the host, and the host is there in the title. Right. So, your E. coli is basically the host, and the glycolysis cycle, the TCA cycles are present within that. Now, there would be a specific gene sequence in the E. coli that would lead to the glycolysis cycle, that will lead to the TCA cycle, that will lead to the other biochemical pathways, right? Once you have identified what each and every genetic material, each and every genetic sequence in a genome does, so you can map the whole biochemical pathway of it. So, if I know that gene A, gene B, gene C, D, E and F, they all are doing multiple things, but they all are interacting with each other. It might so happen that once the gene 3 is activated, that's when only gene 6 will get activated, because it develops a precursor to activate the cycles or the metabolic pathway for the gene 6 or gene 7, right? So, those type of understanding the relationship between the genetic expressions or the expressed products, how they are interrelated, and understanding the whole genome in itself is part of system biology. So, understanding what is happening here is part of system biology, but then you are using synthetic biological tools where you are adding these plasmids. These are the two plasmids here at the bottom. So, you have the origin, and then you are producing your product. You are initiating the production of your product. So, this is carrying your gene of interest, and then it is going to produce your product. And that particular product, the TPA, is going to get released extracellularly, and then you can remove it or recover it. So, now we have moved from a chemical synthesis process into a biological synthesis process. Now, the effectiveness might be comparable, effectiveness might also be less sometimes, but even if it is less, you keep on doing the permutation combination, you keep on running it, you can keep on changing the host, you keep on playing with the feedstocks, you would be able to improve the overall production efficiency of your product at the very end. And this is where I wanted to discuss what the plasmid looks like, but we all were able to answer the questions beforehand, so I am not going to discuss what the plasmid is. But it also utilizes the same design, build, test and learn process. Okay? Now, what are the tools that are available in synthetic biology? So, first thing first would be your engineering DNA. So, on the left on the right hand side you will see in the green. So, you have your engineering DNA, the biomolecular engineering, the host engineering and your data sciences. These are the tools that we basically use as part of the synthetic biology. Now, for the engineering DNA, you are looking at the synthesis. Now, whatever we have discussed till now, even since yesterday, we have been always discussing about making modification to the host DNA, correct? Now, what if I tell you, let's make our own DNA? Can we do that? What is the DNA made of?
Nucleic acids, nitrogenous bases, phosphate group.
And what would be those nucleic acids?
Nucleic acids will be that
Just give me the alphabet.
cytosine, ATGC.
So, DNA is made of A, T and G and C, right? So, it is just a sequencing, right, of A, T, G, C, T, G, A, C, and you can keep on changing it, you keep on getting your double stranded DNA or single stranded DNA. Now, that is the idea. So, can I make it? If I know, if I have, let us say, all my precursors as a building block, right? If I have my A, if I have my T, if I have my G, if I have my C, I can make my own DNA sequence. So, rather than waiting for the host DNA sequence, I can actually make my own DNA sequences. So, that synthesis will require some engineering tools that can be used to do that. Other would be the genome that is present in a host or in the microbe or in the yeast. If I have to sequence it to actually understand what is their ATGC sequences, if I can identify the tools that can be used to identify that, if I can develop the tools that can be used to identify that, would come under the sequencing. You have heard about the Human Genome Project, right? That is where we are sequencing the whole human genome. And in all mystery movies, detective series, specifically, I still remember the one which I used to watch here, CID. I hope you are aware of that one. I love that one, okay, Daya. That one, yes. Okay, so in that, if you have to do the DNA sequence, they do talk about that later in their some of the series, right? So, they are doing the they get the hair sample or the blood samples and they are matching the DNA, right? How do they do that? If you don't know how to sequence it, you won't be able to do that, right? Or you have to put that into a PCR, you get the DNA, you put that into PCR, then you run it, you get the fingerprint, and then you compare the fingerprint with the previous fingerprint, and then you are able to match it. At one point of time in history, that was completely unimaginable. You couldn't even imagine that this thing is possible. Now it is possible. Earlier, the DNA fingerprinting used to take weeks or months. Nowadays, it takes hours because of the engineering advances that have taken place. And those advances that I'm talking about are part of your synthetic biological tools that have been developed over the years. The computational tools, the mathematical tools, the engineering tools, all of this combined have given us the efficiency that we can actually sequence the DNA. So, sequence the genome and get and study the metabolic pathways, understand how each and every genetic code is related to each other and what interrelation do actually they have with their phenotype. You know what the phenotype is? The physical characteristics of it, right? So, how a specific sequence will lead to a variation in the phenotypic pathway, that understanding, that relationship understanding is very, very important when you are working with synthetic biology. Then comes the standardization, the assembly. We can assemble all of these, and even if we want to do the editing like the way we did in the plasmid. So, all of that comes under the tools of engineering DNA. Then some of the examples would be for the synthesis and sequencing. At one point of time, the cost that was involved in terms of looking at in the late 1990s, if you are doing the DNA sequences per bases, the price per base, what would be the base? How many bases are there? Like how many? Okay, what is the base?
ATE is a base pair and GC is a base pair.
So, is A a base?
Yes.
So, for identification of just A, just T, just G, just C. So, the cost per base, not base pair, per base, that is what I was talking about. At one point of time, it's like $100. Now, it's in like in cents, we can do that. So, it's pretty low, right? With the advent of engineering, with the more and more advancement that we have had, this whole thing has reduced. Now, we are capable of doing DNA sequencing and the synthesis at a very low cost. Now, you have the single stranded DNA. If you want to synthesize those, you use a solid phase chemistry, which actually includes controlled pore glass beads, and then you are able to put that, and then you can get your sequences done. People like me, when we do research and if we do need to have a sequencing done, or let's say synthesis done for a DNA, I'll say the one we used to, have you heard about this term? Aptamer. Have you heard this term? What exactly is it? A single stranded short strand of DNA, which might be specific to something specific, right? Like we used to develop these aptamers for mycotoxins. If I have to identify a mycotoxin, if I want to develop a biosensing technique, I would be using these aptamers because they would be very, very effective, and my sensing capabilities would be the best in that aspect. So, to develop these short strand DNAs, rather than you use a process called a SELEX. Okay. Now, that was completely unimaginable at one point of time. So, now it is very, very easy. I used to pay around $40 to get one aptamer done. So, the only thing that I had to tell them is that this is the base sequence that I need, or this is the sequence that I need of my ATGCs, can you give me? And I need a thiol group in the front, I need this in the back, and they will make it, the company will make it and then send it to, they will ship it to us, and then we will use it. It's as simple as that now. Companies like Twist, Agilent, they use silicon chip technologies to make this single stranded DNAs. So, there are different techniques, different processes that are available for you to develop these sequences now. Other would be production of the double stranded DNAs, where you use the polymerase chain assembly for shorter synthetic fragments. And for the double stranded genes are produced from stitching together those short single-stranded oligo using DNA polymerase and PCR. So, PCR, everybody is aware of it. You should have had demonstration. I still remember when I was watching CID at one point of time, they were talking about the PCR machine, and that they showed the PCR machine, but it was a microwave. And he said, like, no, this is a PCR. It does this. And I was so excited that this is how the PCR should look. And when I went for my undergrad, it was a very different machine. But it is fun. Like, you get to know a lot of these things from your day-to-day life, right? So, PCR can be used to make your double-stranded DNAs, and it's a very common technique. Once it came into the market, it has made life much more easier for molecular biologists. Then comes the genetic circuits. Now, genetic circuits is an interesting part. I'm not sure, but have you seen some things like these when you're reading some of these molecular biologies or genetic papers? These diagrams, I'm not saying anything else, but just these diagrams. Now, these diagrams are nothing. This is on the right hand side, you have your code or the language. It's like your A, B, C, D, E, F. So, those are your alphabets. And when you arrange your alphabets, you get your words, right? Similarly, these symbols are used to design your circuit. The idea was that it will resemble an electrical circuit. So, when you have valenic acid and the symbol that is being used is to repress. So, if the valenic acid is present, this genetic sequence is repressed. Okay? So, if valenic acid is present, only RFP is expressed. The rest is repressed. So, the promoter is the start, right? So, then the promoter, this whole sequence will get expressed. So, only RFP will get expressed if the valenic acid is present. So, because it is repressing the rest of the process, the Van R will not get expressed, because you have the promoter here too. But just because of the presence of the valenic acid, it won't work. In this particular case, in the second example, you can see where you have the light source. If the blue light is present, what will happen? Look at the code on the right hand side, look at the diagram on the left hand side, your circuit, look at the code and see what is happening. If the blue light is present, which one will get inhibited? You have the promoter at the very beginning on the left hand side. What is the first thing? YF1, right? Followed by F6J, FIXJ. I'm so sorry if it is not clear. Is it not clear enough? No, the diagram. Is it not clear? Like, are those alphabets legible? No, right? I'm so sorry. So, the very first one is YF1 on the left hand side, followed by fixed J. Then I think it is CDRB, CDRA and CF. Okay? So, if the blue light is on, there is repressor sign. Which one will get repressed? YF1 and so your YF1 will get repressed, even the fixed J will get repressed. With the inhibition of the YF1, the fixed J will get activated. Okay?
Express the CDRAB.
Yes, and if the fixed J is active, you express the CL at the very end. So, that's not CF, that's a very CL. And if no CL, express CDRAB. So, these are that's the language that you are using. So, you are using these symbols on the right hand side to write your electrical circuit. So, this is the very core of synthetic biology, or when you are doing computational synthetic biology, they actually show you these things, and this is something that they will design the genetic circuitry. And you would see the similar example here on the plasmids. You see the promoter on the left hand side at the very bottom. You should see there. So, you have the promoter, and that promoter is going to use and switch on the XYL MSC, and that will lead to the TSA MBCD, and it will keep on going, right. Now, another aspect of synthetic biology is biomolecular engineering. Now, in the biomolecular engineering, what we are doing is the natural macromolecules such as the proteins, they can be engineered to have new functionalities. Similarly, where we were talking about the engineering enzymes, so enzymes are proteins. They are basically used for catalysis, correct. Now, if you want your enzymes to be capable of, let us say, performing catalysis process at a specific temperature. Now, as you increase the temperature, the proteins will get denatured, right? Enzymes get denatured. But what if I want it to be viable at that particular temperature? I can make modifications. I can make engineering like changes into the design of my enzyme itself that it is capable of that particular functionality. So, that will come under your biomolecular engineering. Now, that comes through your directed evolution. I already gave you the answer for directed evolution. So, you make one mutation, you let it continue from one generation to the second generation to the third generation, and then you are able to identify which generation actually has it. Now, mutations, question for you. Let us say if somebody has sickle cell anemia, one of the parents have sickle cell anemia, will the progenies will also get the sickle cell anemia? So, if a parent has that, will the kids also have it? No, every progeny, will it get transferred every generation? It might not get transferred every generation, but mutation which is intentional will get transferred every generation. So, if you want to kill the mosquitoes, make a mutation, the progenies will not, and that is actually a research going on, where they are actually working on changing the DNA sequence in such a way they have put up a mutation, and that mutation, once it gets transferred, it will be transferred from one generation to the second generation to the third generation. But I don't want that mutation, I want it to evolve, I want it to mutate by itself, right. So, that would be the directed evolution. I am directing it. So, indirect evolution is like you just did and you wait, something might happen somewhere. A directed evolution is where you are directing the evolution to go in a specific order. You got the idea? This directed evolution, please remember this term, directed evolution. You might want to use it when we go into the case study too. Just a hint. Now, directed mutation is basically used to make modifications to enzymes specifically. So, if you are making changes to the proteins, to the enzymes, or you are trying to develop new types of proteins, new types of enzymes with different functionalities, you generally utilize the directed evolution process. So, directed evolution is used through rounds of sequence diversification, selections for functions, and amplifications. Now, entirely new protein sequences not seen in future can be generated through the de novo protein design. So, I can actually make my own protein, even that is a possibility these days. So, that will be a de novo protein design. So, something that is not natural, I can make it. Now, another aspect of biomolecular engineering would come with respect to novel metabolic pathways that can be constructed using retrosynthesis approach. Sorry for the slip of the tongue. Now, retrosynthesis approaches, anyone is aware of what exactly it is? It's a very interesting thing, but it's widely used in chemistry. I might be totally wrong. I will never consider myself to be an expert in system biology. I'm an engineer, but this is a topic that you need to have some background in, so that you can understand the rest of the things that we are going to discuss in this particular course. That's the reason why I'm giving you this information. But retrosynthesis approaches in my ideas, let's say if I ask you to make a specific food product, you will ask me what would be the raw material, let's say, right? So, if you have to bake a cookie, you will take the flour, you will take the chocolate chip, you will take the egg, and you take all the utensils that are required, and then you can make or bake a cookie. What if you go backward? Now you know a product. So, let's say I want to have a product A. I know that A can be made from B and C. C is commercially available, but B is not commercially available. So, that means you can buy C as a raw material, but B is not commercially available. But you know that B can be produced from D, but D is also not commercially available, but D can be produced from E and F. F is commercially available, E is not. Okay? But you know that E can be produced from G, which is your commercially available. Now, what are your raw materials to make A? G, F and C. Rest all are not. So, we do not show them as chemical reactions arrows, we always show them as double arrows, because that might be the process that will be used. So, if I have to write it, I will write, so G with some transformation, some chemical reaction gets converted into E, which with addition of F will give me D. Now, this is proper chemical reaction. Now, this D with some chemical transformation will give me B, with addition of C will give me A, which is my final product. Now, this is called retrosynthesis approach. Use that now with your metabolic pathways. So, you know, let's say that acetyl-CoA, have you heard about this term acetyl-CoA? You would have seen it in one of the. Now, acetyl-CoA is also a precursor for many other products. So, you know that acetyl-CoA is one of the process which is like one of the precursors for my particular product, but that is the final product. But you need the cycle for the before you reach that acetyl-CoA. So, you will go backward, you start developing that cycle, and you will somehow put it with your metabolic pathway, your glycolysis cycle, that all this acetyl-CoA will start getting into that pathway, the one of your interest or the one which you have engineered. So, that would be the biomolecular engineering aspect of it. I am not asking you to be a champion in this, I am just trying to give you a basic idea that this is one way that we can do things. Is it clear to you? So, if you want to introduce a new metabolic pathways, you need to know what, like at least one of the components of your metabolic pathway should be present within the one of the metabolic pathways within the cell, and then you can use that as a precursor to develop your own. And then you can use repressors to suppress the rest of the cycle, and all the acetyl-CoA that is being produced will get moved towards your pathways, and your product will only be produced. That is also possible. That is your engineering here. Now, host engineering, we have been discussing this as part of our precision fermentation also. So, what are the different types of hosts that there are, and what changes do we do? So, either we can use a cell-free systems, a single cell or multicellular organisms, or even the biomass or the consortium. So, the changes that we can make. So, what are the different types of hosts that we can use? So, what are the different systems? So, one would be the cell-free system, where you are actually doing it in the lab, in the chemicals itself. So, it can be used as a quick test for gene expression. You grow your compounds to taxene for the cells, and it needs to be extracted and purified. For a bacteria, that's where we get into the fermentation side. So you can grow well in bulk, model for many diseases. We have used bacteria to look out how specific diseases are, how specific diseases can be fixed. Like we can identify the proteins that are eliciting a specific response by your immune system by modifying the host, which is specifically the bacteria here. It's a prokaryotic, can't fold many human proteins. So if you're trying to work with your human proteins in a bacterial cell, it's not possible because it is not capable of folding them. Human proteins are pretty huge too. That is also one of the reasoning. Yeast, on the other hand, is pretty good in that. So if you're going for the proteins and enzymes, yeast is a very good source, or sorry, very good host. So it grows well in bulk, can produce variety of proteins, and often used for food and bread. So in the yeast, the best example for the yeast would be the Saccharomyces cerevisiae. The other hosts that you can choose are your mammals. So you have your human protein folding capabilities that are present because it's us, it's our cells that we are using. You can use that to test the drug therapies. Let me ask you a question. The cell lines that we use, the cancer cell lines, what type of hosts are they? Will they fall within the mammal? The cancer cell lines? Depends.
HEK cell lines and the CHO cell lines, they are the mammalian cell lines actually.
Yes. So I'm just asking, so cancer cell lines, will that fall within the mammalian cell lines?
Yes, sir.
So cancer cell lines, mostly when we are looking at for disease identification of drugs that might act against a specific disease within the humans, we basically use the human cell lines, right? So those mammalian cell lines are basically used. You will appreciate this fact, the majority of the drugs that are developed are proteins. And when you're working with proteins, you always want to see how they will interact. Mammalian cells are best to work with that because those proteins are mostly human proteins. So testing drug therapies, and they are very difficult to grow in bulk. I have my own personal experience during my master's where when I was working on the cancer cell lines, they will die multiple times. I have to repeat that same experimentation like 6 or 7 times to be able to do it. A small modification, like even like if I'm moving it from one location to another location, I basically just added a bit more force, I'm done. Restart the whole thing. It used to be fun, but not interesting. That's why I moved back into engineering. But anyways, plant cell lines, they are good for the photosynthetic pathways, and if you're looking at varieties of compounds, if you're looking at generation of phyto compounds, then plant cells are very good hosts. Very few tools are available to work with them the way we have it for the mammals, for the E. coli and the yeast. There are so many genetic and engineering tools that we can use. For the plants, we don't have that many. It's not a very well studied area. It's also very difficult because of the presence of the cell wall. It creates another hindrance. Whole organisms, that's when we go for the mouse model, and I'm going to now question your ethical aspect here. How many of you have actually worked with mouse models, if I may ask? Have you ever seen a mouse model, like white mouses, the ones which are used? You know how we have to extract our things out of them, right? What do we do?
Sir, dissecting, like extracted the PBCs, peripheral blood cells,
Yeah.
out
So
of it.
how did you do that? So
Sir,
before that, the mouse was alive.
Yes,
You had to kill it.
yes, sir.
So that's
Sir,
where the ethical question comes in picture, right? Whether you do it, whether you not do it, then it's a humane way, non-humane way. I remember when I was doing my master's at McGill, one faculty in molecular biology and microbiology, he taught me how to do it. I was a TA for a course in microbiology, and then we had to demonstrate to the undergraduate students how to do that. And oh my God, like the students will cry when we are doing that. Even we used to cry. So that's one, for me personally, it's an amazing thing to work with them, but that's ethical aspect comes into picture. So that's why you will see majority of researchers are trying to identify hosts or identify processes in which they don't have to go into this, but sometimes it is necessary. Because when you want to see the phenotypic differences, right, that's the only way you can see it. You cannot see the phenotypic differences in the microbe, but you can only observe that in a living organism, and that would be your mouse. And it's most difficult to engineer at the cell level. Now comes the end of your synthetic biology part. Now we have been discussing about the cells, we have talked about the DNA sequences, the genetic sequences, the proteins, directed evolution of the proteins. Now the question for you would be, what sort of information do I get from synthetic biology? Like, what are the techniques that we are using? Everything that we talked about, they can be termed under an umbrella of something called as omics, right? Proteomics, genomics, metabolomics, transcriptomics, and please go ahead. Majority are these. So they will fall under the omics that develops the data that you get. Now from the proteomics, you can get the information about the protein. So if, let's say, if I ask you to give me the structure of the protein, I give you the protein name. If you have to identify the structure of the protein, where do you go? Where will you look for the structure of the protein? On NCBI PDB, the protein database bank, right? So that would be the very first place I will look for a protein sequence, right? If I want the sequence and then the 3D structure or the 2D structure, I will be, or the primary structure of the protein, I can get all of that information from the PDB if it has been crystallized and uploaded. Not all proteins, we have not been successful in crystallizing all proteins yet, but that's an ongoing process. As people are doing that, they keep on uploading on the PDB. So you will be, that will be your very first source of information. What if you want to study how the evolutionary trend of genetic sequence or the mutation took place? What will you use? Or what do you want to understand the homology between one sequence and another sequence? If I remember correctly,
tree diagram.
Yeah, it used to be BLAST, FASTA,
Yes.
all the bioinformatics tools that you can use, right? Application of that also falls under synthetic biology. And you have to excuse me on that because I am not an expert in bioinformatics, so I cannot teach you BLAST and FASTA. I would have done that as part of my undergrad, but not when I am doing my research. I am not working on these areas. But these are the tools that are available. Now these will provide you the data that you can basically analyze later on to understand the metabolic pathways. So if you get all the omic data, you can understand the metabolic pathway. You can study each and every sequence of the genome and understand the relationship, the interaction between the different sequences. One metabolic pathway and other metabolic pathways, because sometimes the product of one metabolic pathway is the precursor for the other metabolic pathway. So understanding that relationship, having that as a map will help understanding and developing new strains, developing new methodologies to develop or to produce your product of interest using precision fermentation. So the key takeaway is the diagram itself for your synthetic biology. We will take a break, short break, and we'll come back later.